My name is Emma and I'm one of the scientists here at Cellomatics. I did a degree in biochemistry at Nottingham Trent University and then I did a PhD at the University of Nottingham. My PhD focused on the use of human-induced pluripotent stem cells and CRISPR-Cas9 to model extracellular matrix components and their role in a set of diseases called lysosomal storage disorders. My main interest at the time was just working with human cells and cell culture, because I'd done a project during my undergrad which was using mouse cells and culturing those, and I really found that interesting. At the time, CRISPR-Cas9 as well was relatively new, so I found that aspect really interesting, and that's something I wanted to do as well. I wanted to come to Cellomatics because I like that all of their assays are cell-based and it involves a lot of cell culture. That's something that I found really interesting and I think it's important as well. So Cellomatics is a CRO which specialises in performing and developing in vitro cell-based models, and we focus on 5 main therapeutic areas, which are immunology, immuno-oncology, oncology, respiration and inflammation. And since I've been here at Cellomatics, I've mainly focused on the respiration and inflammation therapeutic areas. And these therapeutic areas have involved the use of primary human cells quite a lot. And it also involves some of our more complex transwell-based models. Transwells are a permeable membrane support that allow access to both sides of a cell monolayer, but they also aid with multicellular setups as well. Transwells sit in a plate, and usually you see cells in the apical chamber of the transwell, or what you would consider the top of the transwell, and we usually fill the basal chamber with media as well. Cellomatics use transwells for a lot of their models, for example our COPD model. Our COPD model uses bronchial epithelial cells, which we seed in the apical chamber of the transwell and fill the basal chamber with media. We can then remove the media from the apical chamber and rely on the basal chamber to provide the nutrients to the cells. This is what we would call air-liquid interface, and it better mimics the in vivo environment a lot more, especially in the lungs. We also use transwells for our blood-brain barrier, or BBB, and our asthma model as well, which are both tri-culture setups. Another key disease area that we use transwells in is inflammatory bowel disease. Inflammatory bowel disease is a group of chronic inflammatory diseases that affect the gastrointestinal tract. The 2 main types of inflammatory bowel disease, or IBD, are Crohn's disease and ulcerative colitis. IBD pathogenesis is caused by lots of different factors, such as environmental elements, genetics, microbial flora, and the immune response as well. Often IBD is as a result of a complex interaction of all of these different factors together. Cellomatics has lots of different models to study IBD. Our most common is the use of CACO2 cells, which are derived from a colon adenocarcinoma. In this model, the CACO2 cells are seeded into the apical chamber and allowed to differentiate into a polarised monolayer. The polarised monolayer consists of cells that express tight junction proteins, and these tight junction proteins are really important in inflammatory bowel disease because they're what determines the integrity of the intestinal barrier, and often in IBD patients we see a reduction in these tight junction proteins and also an aberrant immune response as well. So once the polarised monolayer is formed in the transwell, we can treat the cells with reference and client compounds, and then we stimulate the cells with IL-1 beta and LPS to induce the IBD phenotype. We can grow the CACO2 cells as a monoculture, but we can also combine them with THP1 derived macrophages or freshly isolated primary human immune cells from either diseased patients or healthy patients. And as well as CACO2 cells, we can also use primary intestinal epithelial cells, either with or without the immune cells. We stimulate the primary cells in the same manner as the CACO2 cells, and then we can collect either the supernatant or the cells for downstream analysis. As I mentioned before, the tight junction proteins are a really important part of IBD and the barrier integrity. So we can actually measure the barrier integrity of our intestinal barrier within the transwell by determining the transepithelial electrical resistance, or the TEER. To measure the TEER, we put 2 electrodes into the transwell and we run a current between them, and effectively the measurement of the TEER is whatever current that the cells are soaking up themselves. So a higher TEER would mean a more integral barrier and a stronger barrier. We know, in our model, we note a marked decrease in the TEER readings following stimulation with IL-1β and LPS, and we see a rescue of those TEER readings following treatment with our reference compound. As well as the TEER, we can also measure tight junction protein associated genes, including ZO1 and occludin, and we can measure these by qPCR. Here at Cellomatics, we have a Quant Studio 3, which allows us to plex up to 4 genes in a single sample. Similarly with the TEER, we see a decrease in the tight junction protein associated genes following the stimulation, but we see a rescue of this after we treat with our reference compound. We can also run a Luminex multiplex assay using the cell culture supernatant. The Luminex multiplex assay allows us to plex up to 50 cytokines within a single sample. Usually for IBD, we measure IL-8, IL-6 and TNF-alpha, but this is completely customizable. If the immune cells are of interest, we can collect the supernatant from these and run a similar cytokine assay to determine how the cell-to-cell interaction is affecting the immune cells. It's important to mention that all of our assays are completely customizable, so with this IBD model that I've just mentioned, we can customize the treatment time, the stimulation time, the readouts, the genes of interest, the cytokines of interest as well, and it's all completely down to the client, including whether they want CACO2 cells, primary cells, or whether they want that immune component as well. While at Cellomatics we have quite a vast knowledge and expertise surrounding IBD models, we do try and advise the client on what can best suit their needs, but the final choice is down to them. So current therapies and current research is focusing on anti-cytokine antibodies, but again, these have worked with varying success. So because of the varying success with both established and new therapies, the need to find something for IBD is ever increasing, and Cellomatics can support you on your drug discovery journey, particularly with IBD. So if you're interested in anything that you've heard in this podcast or would like some more information about Cellomatics, then you can either visit our website or email us at info@cellomaticsbio.com.